is a work that David Stokes and I started about 3 years ago, with the realization that we all had samples, eukaryotic membrane protein targets and prokaryotic membrane protein targets that we wanted to evaluate for crystallization. And we wanted to, we all had been doing it manually and on a smaller scale, and we wanted to be able to explore more the two-dimensional crystallization space of our targets. As this is a relatively current table of all the membrane protein structures solved by electron crystallography. The ones in bold are solved to near atomic resolution, and well, you can see that indeed you can solve membrane protein structures by electron crystallography. So the technique is sufficiently mature to allow you to do that. There are several limiting steps, and certainly one of them is the ability to obtain, well-diffracting 2D crystals that are not only diffract well, but also are going to be good specimens for data collection. So some of the advantages of 2D crystallization and electron crystallography is, of course, that the membrane protein can be studied in its bilayer environment. The 2D crystals perhaps can accommodate conformational changes a bit better than 3D crystals can. And of course, if you are interested in studying complexes, the 2D crystals both phases are fully accessible to the aqueous medium. But on the other hand, screening of 2D crystallization trials is slow, and obtaining optimal crystals for the structure determination is difficult, and they are relatively rare. Data collection is tedious. 2D crystals are flimsy, so even if they are good crystals, they might not behave as optimal specimens for EM. And then, of course, sometimes some of the maps suffer from anisotropic resolution. So these are some of the things that I believe that we need to be able to, me and the rest in the field also believe that are the hurdles and bottlenecks that we need to overcome. This is just a general strategy for 2D crystallized membrane proteins. We are first going to express a target of interest, in this case this protein in gold. We are going to most likely solubilize it in detergent out of the membrane. We are then going to purify it in that detergent, and we are going to provide exogenous synthetic phospholipids or from natural sources. We are going to remove that detergent out so we can get a 2D crystal which, as you can see, reflects pretty much the original status of the protein. And once we do 2D crystallization, we need to image these outcomes. So my talk today is going to be about how to streamline many of these steps. And for that this is our current 2D crystallization pipeline. We collaborate with NICOM's New York Consortium for Membrane Protein Structure, where they express and purify membrane protein targets of several sources. So we have a constant supply of membrane proteins. We then carry 2D crystallization in parallel on 96 well plates, so we can screen 96 conditions at once. Because we have 96 conditions, we have to now prepare specimens from 96 from each of those conditions, and we need to do that also on a 96 well format using a magnetic tray and using a liquid handling robot. And then we need to now image each of those grids, each grid representing one condition of crystallization using an automated loader and an automated imaging system. And then we need to do data analysis. At the moment, this is done by an expert microscopist. Ideally, we should have algorithms that are able to recognize 2D crystals, not only when 2D crystals when the conditions have already been optimized and they are easily recognizable, but really trying to recognize them already at the stage of screening. This is much more complicated. This is a table of targets that we have tried during the last year and a half. These are 15 different membrane proteins, most of them from prokaryotic sources, although we also have here a GPCR. Many of them are intramembrane proteases. This is just because this is one of my research interests. They are from different organisms, and they have a variety of functions. If you look at the molecular weight, we have, they range from about 20 kilodaltons all the way to 160 kilodaltons. But most of them are relatively small membrane proteins. The PIs are relatively normal. If you look at the number of transmembrane domains, we go all the way from 3 all the way to 17, but mostly are membrane proteins that have between 3 and 6 transmembrane domains. Most of them have the N-terminal inside, and some of them have have it outside. Some of them are more hydrophobic than others, like not so hydrophobic. So you can see that we have here a variety of targets. I'll tell you in my last slide what is the success rate for all these targets. So these 15 targets have been optimized for purification. They can be expressed in large amounts. We need about 3 milligrams of protein currently to run 100 conditions, so it's still a very substantial amount of protein, certainly compared to the current amount of protein that you need for 3D crystallization. So they are all relatively pure. This is something very important that we do. So you need to you are going to work in detergent, your protein is going to be purified in detergent, and certainly at the earliest stages of crystallization, you're going to be in mixed micelles, so your protein better be stable in detergent. What we do with each membrane protein is we do a very stringent test. We take each membrane protein, we purify it most of the times in dodecyl maltoside, and then we want to know whether the protein is stable on that detergent. For that, what we do is we incubate the protein with DDM, LDAO, C84, and beta-octyl glucoside. These are common detergents used in X-ray crystallography. Some of them, like LDAO, might be a bit more denaturant than the others. And what we do is we incubate the protein on a concentration of detergent that is 5 times the CMC. We incubate the protein for 1 hour, and then we run it through a gel filtration. And then we evaluate the peak. The sharper the peak, the better, and we evaluate the position of the peak. We then leave that membrane protein 24 hours in that detergent at room temperature, and then we rerun the experiment to see whether the protein behaves exactly the same way. If a protein behaves like this target here, stable in all 4 detergents, monodispersed, very sharp peaks, chances are at currently about 50% that this protein will crystallize in 3D. And about 80% of the times this protein will crystallize in 2D. So really starting with a protein that is very stable in detergent is key. It's also very important to determine how much detergent your protein is carrying with it, because this is the detergent that you are going to have to remove. And remember, your membrane protein, really the concentration relative to the total volume of the sample is not important. What is important is the concentration relative to the total volume of micelles that you have in that sample. So you will have 10 micromolar protein concentration. If you increase the detergent concentration so much, the protein will be, even if the volume is kept constant, the protein will be completely diluted. You might lose oligomeric state, etc. So determining detergent concentration of the protein is very important. You can either use colorimetric detection. A simple TLC will carry a long way and will help you to determine the concentration of almost all detergents. And following a bit on the footsteps of Andreas and the people in Basel, we are now also currently use drop shape analysis to determine detergent concentration in our samples. Once we know the detergent concentration, we know the protein is stable in detergent, we start setting up crystallizations. We do that currently on this type of device, which is basically a 96-well plate that allows you to have 96 samples dialyzed against 96 independent buffers. The volume that currently we use for the sample is 50 microliters, and it can be dialyzed against 1 milliliter of sample. These are our standard conditions for initial screen. We use all the usual suspects of lipids: DMPC, DOPC, E. coli lipid extract. We do the screening at 3 different pHs, 6, 7, and 8. 3 LPRs, lipid to protein ratios, to begin with, and then we use just normal cations. With all this, combination of all these parameters already gives you 96 conditions. Now, you will have to set up 96 dialysis buttons and set up and prepare 96 specimens and image them manually. Even a very motivated and talented postdoc will run out of hope very fast. So you really need to automate all this. Something else that we do is we need to, because we know in which detergent the membrane protein is stable, and the next steps for us are really to measure, not really in which detergent the membrane protein is stable, but whether it's more stable in one mixture of detergent mixes than detergent lipid mixes than others. So determining the stability in mixed mixes is more important, I think, than determining the stability in detergent. But we need to make sure that we have mixed mixes in the detergent of interest. So what we do is just by measuring absorbance at 500 nanometers and measuring basically the scattering of vesicles, we can determine the minimum detergent concentrations in these scales for OG that we need to solubilize each of the lipids that goes into the screen. So really, again, emphasizing that reducing your amount of detergent is very important. We all concentrate membrane proteins before crystallization. That, of course, leads to increased detergent concentrations. You really need to make sure that you know your detergent concentration that is kept at a minimum. We then have 96 after the crystallization. The crystallization can take up to 2 weeks. And the dialysis block is able to dialyze DDM and other low CNC detergents by doing several exchanges and so on. So that works well. We need to prepare specimens, and we do that in parallel too, using a magnetized tray. These trays are commonly used for high throughput labs to purify protein by magnetized nickel beads. So they were available. And they fit very well. They can accommodate a nickel grid very well, and the nickel grids hold tight to the magnet, and so it's very easy to deposit the 96 grids. We then use a liquid handling robot to apply sample 2 microliters of each of the condition, and then to stain it. The trick on staining is to use a very low concentration, 0.25% of uranyl acetate. In our hands, if we go higher than that, we get a lot of precipitation. And then we can just, by taking a long strip of filter paper, we can, in the last step, if needed, we can blot 1 row of grids at a time. And once you have 96 samples, if you want to image them manually, remember each of the grids has to go through the airlock of the microscope. You need to previously align the microscope. You need to go through the airlock. You need to find out regions of interest and so on and so forth. So if you do that on 96 grids, it's really very time-consuming. So you need an automated imaging system, and there are several versions of such system. One system is the one that Angie and Bridget Carragher use at Scripps. Another version is the one that Andreas and Henning have, I believe, here. Another version is the Gatling gun that Subramanian, in collaboration with Gatan, developed. And this is just a 4th version of such a system. Basically, we have a screening microscope, and then we have 2 robots, one a SCARA robot. Both robots are made by established manufacturers and are used commonly in industry. This SCARA robot can basically rotate, has several axes of rotation, so it can do all sorts of movements, and is the robot that is in charge of picking up the specimen of picking up the grid and putting it on the sample holder. And this Cartesian robot is very simple but very stable. Will pick up that holder and will bring it here and will just insert it into the microscope. So this is just zooming in a bit on the SCARA robot from Yamaha. Basically, each of the grids is deposited on this anodized tray. It's anodized because this is the tip of the robot. The robot has 3 nozzles. The nozzles contact the rim of the EM grid and they apply vacuum, so the grid is picked up by vacuum. And we have a laser here that determines whether the grid has been loaded or not. And that's why we need it. That's why we need an anodized tray. Then for loading, we modified the sample loader. We modified the tip of the sample loader basically to have a lever, a clamp that will hold the grid. And this is, again, the tip of the SCARA robot, and we just attach an Allen key here. So now the robot can basically go underneath this clip, can lift it, then deposit the grid, and then close it very easily using the Allen key as a hand. Both the SCARA and the Cartesian robot are controlled by a program called iRobot, and the entire process is controlled by Leginon. So we collaborated with Anchi and with Bridget for them to help us integrate their software in with our microscope, and also to integrate their Leginon had already embedded an automated software, so we just integrated it with the GOL. So Leginon now will control these 2 robots, will control the automated imaging, and will also send all the images to a database, to a laboratory inventory management system, which I will show you a bit later. This is a movie of the robot. So the SCARA robot is now going to open the clamp. It's going to pick up a grid. Places the grid, makes sure that there is a grid in position, and closes the clamp. The SCARA robot is now finished, goes into a resting position, and the Cartesian robot picks the sample holder. And inserts it. So it has taken us about 1.5 years. The robot came into the lab about 1.5 years ago, and it has taken us 1.5 years to have it up and running and to solve all the bugs and all the problems, but now it's really running quite smoothly. With the imaging system that we have to evaluate, to load and evaluate 25 grids, 25 grids at the moment, it takes us 18 hours. So we need almost close to 3 days to evaluate a full screen of 100 grids. And as I said, the imaging is controlled by Leginon. And this is a bit, the scheme that we use for imaging. We do a montage, an atlas of the grid, 3 by 3 in this case. We take low images at low magnification. We used histogram criteria to find the different grid squares and to select grid squares that either are completely black because something went wrong, or the stain is covering the entire grid square, or the carbon is broken and they are therefore completely clear, or basically the edge of the grid square is in the middle of the image, and therefore you should not take any images in that region. And then automatically the software selects regions of interest within the grid square, also again using histogram criteria. And these are basically the outcome types of images that you should expect, either some crystals, some helical crystals, vesicles, or other things. Once you have finished imaging the entire each of the conditions, then you are going to have to evaluate it. And at the moment, this is done by a postdoc. So we have 6 types of objects that you are going to find on the grid. First, you are going to find most likely a lot of macroscopic precipitation. This you will not find on an EM grid. Each of those conditions, we look at it under the light microscope as you would do if you were a 3D crystallographer. We often find that the EM grids are completely clear, no sample whatsoever, and when we look, everything is here. So large precipitates will not deposit on the grid. So you will get confused. You need to look under the light microscope. Many of the membrane protein targets that we are working with, they are very stable, and they are also 3D targets of us. So many of them give us macroscopic precipitation. Many of them give us microcrystals. Then we are going to find some lipidic structures, which we don't know exactly what they are. We call them lipidic structures. They are very rich in lipid. They are somewhat vesicular. You know that lipids have a lot of polymorphism, so we also find aggregates, of course, and then if things are going well, you start to see proteoliposomes, perhaps parallel edges, and if everything goes really well, you get a crystal lattice. And this is just how we evaluate currently our screen. We basically assign, based on this criteria, we assign we assign values depending on how abundant each of these features is in the grid. So number 4 will be like in here in DOPG for this protein at 0.5 LPR. We are finding a lot of crystals in which the crystal lattice is either clearly visible or if you do a Fourier transform of the image, you get diffraction spots in negative stain. Then Legionor automatically interfaces with this program. It's a laboratory information management system. This program is called Sesame. It was developed by Sol Knight at the University of Madison, Wisconsin. Basically, these programs were initially developed for structural biology programs, for structural genomics. X-ray crystallographers were generating humongous amount of data, and maybe one lab was purifying the protein, sending it for crystallization to another lab, and maybe some third lab was doing a structure determination. So they needed a program that would allow you to interface between labs. And so in this case, in Sesame, we load the target, we load everything that is associated with the target and SDS page, protocols, etc. And then we also load the conditions. Because we mostly use similar conditions for crystallization, you only need to load them once, and then you can copy them to your other targets. And then these images of each of those conditions, in this case, this particular condition, this is very different from the screen that I showed you at the beginning. This is part of an incomplete factorial screen that we used, developed by Michael Landsberg here, and by Ben Hammer in Australia. And we were using their screen to see whether we could improve our 2D crystallization success rate. So each of those conditions will now have images associated with it. So we manage the entire system this way because we've done around 50 different screens this year, 50 screens, 100 conditions each, so we have about 5,000 grids to evaluate, so that really multiplies into thousands of images. So after a long story, how did we perform? Well, you can see that this is the best outcome that we obtained for all these membrane proteins. We obtained diffracting crystals for 3 of them, and we obtained sheets and tubes with parallel edges to many of them, for many of them. So in fact, the success rate in obtaining either sheets or tubes is very high. The success rate of obtaining so far diffracting crystals is about 30%. The success rate of getting diffracting crystals plus sheets and tubes is about 80%. In very few cases, we get aggregates, like here with this particular rhomboid. So either because the proteins have been optimized and are good expressors and are stable in detergent, or either because the screen is able to cover substantial amounts of crystallization space, we get a relatively high success rate. Many of these are now, we are rerunning screens to improve the diffraction quality of these crystals, because of course that is the next step. First is to find, the conditions where you can potentially obtain good crystals, and then to really improve them and to make them very large and optimal for structure determination. That's the next step. Something that we want to do in the future is follow a bit on Remigi's steps. They, together with Andreas, they developed this 2D crystallization method, which is based on an old idea that you could basically remove detergent from mixed micelles by complexation to cyclodextrins. And we've been using, Remigi used this method to obtain 2D crystals, and we are using it in the lab quite successfully. We have now characterized the system. We know which the concentrations and the types of cyclodextrins that we need to use, and indeed we obtain 2D crystals with our 2 or 3 of our targets. So the idea, what I would like to do in the future, is to minimize the time that the protein is in mixed micelles, because that is really the time in which the protein is most vulnerable. You need to get it into liposomes as soon as possible. So I would like to accelerate this process using cyclodextrin, and then to allow some time for the proteins to diffuse laterally and to interact into 2D crystals. So our next screens are going to be basically, we are going to use the same dialysis devices in which we are first going to complexate the detergent away, and then we are going to allow the protein time for crystallization. And as we move to eukaryotic membrane protein targets, we no longer have 3 milligrams of protein. So we have, in collaboration with GM Biosystems, we have developed it's not nano, but it's a micro dialysis device, which now only uses 5 microliters of sample instead of 50 microliters. And basically, this is each of the dialysis wells. This is a cross section. So you put the sample here. 5 microliters is sufficient. You have a dialysis membrane here, and then the rest you fill up with your buffer of interest. Now, with this device, and we know it works, we have tested it, we can now test 100 conditions with 0.3 milligrams, so basically 100 times less protein than, 10 times less protein than before. And this is just 30 seconds. We very recently got this project funded, the Transcontinental EM Initiative for Membrane Protein Structure. It's transcontinental because it involves several groups across the US. This is a U54 center funded by the NIH. The NIH, the Protein Structure Initiative of the NIH, has realized that they want to move more into membrane protein biology. We competed against X-ray crystallographers and NMR spectroscopists to get this, and we were funded. All this is spearheaded by David Stokes, is the PI, and there are another 5 labs, Andreas, Tamir, James Love, Pavel Pensek, and myself involved in this. This is a 5-year grant where we want to really push structure determination by electron crystallography. And I'm very happy that, for example, in this case, Pavel Pensek, one of the developers of Chimera, will be working with us to try to use single particle averaging techniques to improve the quality of crystals. And the goals of this grant is to solve structures of eukaryotic membrane proteins, to study their molecular mechanisms in lipids, and to accelerate membrane protein structure determination. And we currently have, the grant will start in a month and it will be run for 5 years. We currently have multiple positions available for biochemists, electron crystallographers, and software developers. And you can look at those positions here. These are everybody involved in this project. Very important, David Stokes, and the people that David and I share at the NYSBC, 3 postdocs. Martin is instrumental in running all our operations. We also get a lot of support from the staff members at the NYSBC. The people at Scripps helped us with Leginon. We get proteins from NICOMs. These 2 engineers built the automated imaging loader and imager. We collaborate with SALT on Sesame and with GM Biosystems to develop the dialysis plates. Thank you very much for your attention.
The way that you were scoring your trials, you had a whole bunch of representative images up the top, and you were indicating when you had proteoliposomes, sheet, tubes, and this sort of thing. So proteoliposomes, and then you showed all the results that you got with your protein. You had a lot that had sheet and large proteoliposomes. Is there any discrimination between your range of the protein within those? Were all of those proteoliposomes that showed ordered to the array, or some of those sheet-like structures that didn't have order?
No, so many of all those that showed sheets or tubes, most of those showed either tubes with parallel edges, but no diffraction.
Okay, so to follow up on that question then, what has been the follow-up success rate with optimizing those conditions to yield diffraction quality crystals?
Yeah, so we, at the moment we are optimizing 6 of those candidates, and the success rate of optimizing from tubes to diffracting tubes is about, of those 6 we get 2 so far in the diffraction. So to assemble the dialysis block, you can either do it manually with a multi-channel pipette, and then it takes about 10 minutes to load, or you can use a liquid handling robot. But we do it manually with a multi-channel pipette. And afterwards, the blotting is also manually? No, the blotting is done with a liquid handling robot. And that works? But you know, Angie already has a paper in 2007 where they used a liquid handling robot to do the negative staining. You don't blot. What you do is you apply a stain and then you remove. You apply, you remove, and you need to have, low concentrations of uranyl acetate and a cycling, an order of events that avoids precipitation. But often we don't even have to do the blotting. That is done manually. So the grids are sitting on these magnetic trays. You need to take them one by one and put them on the tray. And then you leave it for 3 days. And after 3 days, you have some hundreds of images that you will have to evaluate. takes about 10 minutes. 10 minutes to exchange grids. We realize that for that, the vacuum of the microscope has to equilibrate and so on and so forth. So the robots are fast. The limiting step is how fast you can acquire vacuum after you break the air lock. So we think that to evaluate each grid, it takes in the order of 20 minutes. So as I said, so it's basically 25 grids in 18 hours. So to evaluate everything, for Legionel to find out the exact regions of the grid where it wants to take images, and then to take images and all that, it takes about 40 minutes.
Yeah, the evaluation by the person typically takes just an hour. It's the acquisition time and other things that
yeah.
Can we move on?